So it's my pleasure to introduce our next speaker, Andrea Carfi. Dr. Carfi is the Chief Scientific Officer for Infectious Diseases at Moderna. He holds a Master's of Science in Chemistry, a Master's of Science in Physics, and a PhD in Biophysics. He trained as a postdoctoral fellow in the laboratories of Don Wiley and Professor Stephen Harrison at Children's Hospital in Boston. He's an author on more than 100 publications, and he is currently a leading scientist working in infectious diseases and research and early development and Moderna. Of course, he has been, I'm sorry, I'm kind of skipping around to, he's been in research and development for over 20 years at Moderna, GSK, and Novartis and Merck. And it's just a pleasure to have you. Thank you so much for joining us today.
Thank you very much for the... So thank you very much for the introduction, and thank you, Bali, for having me here today. It's great to attend a meeting on vaccines. I think it's very important to talk about vaccines right now. And also thank you, Sarah, for walking us through what it takes to be ready for the next pandemic and all the work that it took to really address the SARS-CoV-2 pandemic 2020. So what I will talk about today is, use of the mRNA technology, and specifically mRNA vaccines for respiratory vaccines and some of their combinations and some of the work that we are doing at Moderna. These are my disclosures. And so you should be all, I think by now, everyone is familiar with the technology that basically consists in the delivery of messenger RNA to the cells. mRNA is embedded in lipid nanoparticles that are composed of PEG lipid, ionizable lipid, and cholesterol. And once the mRNA LNP is taken up by cells, mRNA is released from endosomes. It's taken up by the ribosomes, and that results in expression of different proteins, whatever is included in the open reading frame of the mRNA. And this allows us really to express many different kinds of proteins that can be soluble, secreted, membrane-bound, or intracellular. And of course, in the context of vaccines, generally we express a protein or multiple proteins from a pathogen. Of course, this is a platform technology. So really, if you want to change the antigen, for instance, if you want to update your vaccine in the context of seasonal vaccination, the only thing that you have to do is really change just the sequence of the mRNA. Everything else, the lipid nanoparticle, the reagents, the process remains the same. And so this allows really for really fast manufacturing and development. So we have been using mRNA LNP, of course, for developing different vaccines. And here it's really, there was some of the work that we did during the pandemic. I want to point out a couple of things. First of all, just because of the work that we and others together with NIH and other academic labs had done in the context of the design of vaccines against the MERS and using either proteins or mRNA, we were able to really have lock our antigen design within 2 days. And within 1.5 months, we were in a phase 1 clinical study. And this, of course, was a really fast development, unprecedented. And within 11 months, before a year, the vaccine had emergency use authorization in the US for, of course, prevention of COVID-19. During this time, we also, of course, introduced digitization within the company, AI, and also invested heavily in manufacturing in the US and outside. So, of course, this was also possible because of the strong collaborations with the academic institutions, NIH, and the government funding, and really collaboration across all the companies in this situation. So since then, we've been developing a number of different vaccines, and I would like to just basically in 4 main areas. Today, I will talk about the respiratory and the seasonal vaccines, but also we have developed vaccines against latent infections, such as herpes viruses. We also have targeting other pathogens. And then outside the infectious disease, we are also using the technology to develop therapies for rare disease and oncology therapeutics. And maybe this will be a topic for future talks, but we are getting very exciting data in both rare disease and oncology. So between 2021 and 2023, we really worked on developing the pipeline, especially in the respiratory side. Between 2023 and 2026, we are really focusing on getting this vaccine out for approval and then commercial. And of course, after 2026, we will see also hopefully approval of additional vaccines. And so where we are today, you are all familiar with the Spikevax. That was the mRNA vaccine against COVID-19. Since then, we have developed an improved version called mRNA Spike, mRNA 1283. I will present some data today. We have developed a vaccine against RSV, mRESVIA, that is now approved in the US, and also a version of the vaccine for the 18 to 59 with underlying conditions. The next wave of respiratory vaccines are flu. This is a seasonal vaccine that I'll present some data, combinations. And then, as I mentioned, after 2026, we hope to bring to licensure some of the other vaccines against latent infections, such as EBV, but also GI tract infection like norovirus. So now I'll move to our work really in the respiratory portfolio and really in the COVID-19 space. And I'll describe work that we have done on the next generation vaccine, mRNA 1283, but also the work that we do every year to update the vaccines for new strains that are circulating. So the initial vaccine that was developed is 1273 and was encoding for the full spike protein. However, even during the pandemic, as we didn't know if this vaccine was going to be efficacious, and also as we wanted to develop something that was more thermostable, we worked on a shorter version of the vaccine that we call mRNA 1283. This encodes for two domains, the N-terminal domain and the receptor binding domain of the protein, connected through a linker and with a transmembrane region. The NTD and the RBD are the key targets for neutralizing antibodies, and this molecule, again, is smaller than the full spike. So what that allows us to do, well, first of all, we saw that this molecule, probably because of the smaller size, has higher expression, almost tenfold versus the 1273 version. And also it's more thermostable. And this is shown here, where the 1283 version has a higher stability than the 1273. And this is due to the size of the mRNA. The mRNA is a very unstable molecule and can be hydrolyzed and degraded. So this seemed to provide some potential advantages, and we tested it in a phase 1 clinical study. And here in the phase 1, we compared 1283 at different doses with the 1273 at the approved dose of 50 micrograms. And if you really look at the titers, even with the 2.5 micrograms and compare it to the 50 microgram 1273, we saw comparable or higher neutralizing titers. So the increased expression and the focus of the response towards the critical domains allows for dose sparing. So this program was moved to a phase 3 clinical study. We performed a randomized blind active controlled phase 3 trial comparing 1283 at 10 micrograms, encoding for Omicron BA4, BA5, so a bivalent vaccine, to 1273 at 50 micrograms, the approved dose. Roughly 6,000 subjects per arm. And really we looked at the safety reactogenicity, comparing the two, non-inferiority immunogenicity, and non-inferiority in efficacy. And here just quickly to point out, these are really the systemic reactions, and light blue is 1283, dark blue is 1273, and you can see that really in terms of systemic reactions, the two vaccines are very comparable. So then we looked at efficacy, and we were pleased to see that the point estimate efficacy point estimate was 9.3%. So the vaccine was non-inferior versus 1273 and suggesting, trending towards superiority. We compared also across age. And what we saw is that if you go to 50 plus, so more than 50 years and then more than 65, the efficacy becomes even higher. So this vaccine seems to provide additional protection, especially in the elderly versus the 1273 vaccine. So what about, so in the trial also we had a number of severe cases, 55. This is the CDC definition. And so we could compare again the two vaccines. And what we saw, although they, of course, the error bars are larger, in this population, so in prevention of severe disease, the vaccine efficacy is even higher. What is, what about now the neutralizing responses? So we compared again 1273 to 1283 and in a different age cohorts. And you can see that in general, even with in the 65 plus, the 1283 has higher neutralizing responses than 1273. So neutralizing responses basically trend with efficacy, suggesting the neutralizing responses are actually a correlate of protection. So we also looked at the durability of the response. And again, in dark blue is now 1273 and the light blue is 1283. And what we can see at the peak, of course, we have, as I mentioned before, higher titers for the 1283. The decline in antibody response that is expected for any vaccine after this boost is the same. And so titers remain higher for 1283 after 6 months. I want to point out that these titers are basically above the titers that you see for 1273 after 3 months, and suggesting that this vaccine might provide a longer duration of protection against COVID-19. So in summary, for this part, the vaccine matched the specified relative vaccine efficacy, the objective of non-inferiority, and also we had higher point estimate, especially in the 65 plus. In terms of immunogenicity, of course, we also met the specified objectives, and the 1283 vaccine had higher neutralizing titers than the 1273. In terms of tolerability, as I mentioned, the vaccine, I didn't show that, but the vaccine 1283 had trended towards a better local tolerability, but certainly in terms of systemic reactogenicity, the two vaccines are very comparable, and there were no safety concerns identified. So now I'll switch to what we do to be ready every year for new strains, so for updates to the vaccine. And this is a really working on seasonal vaccines that takes additional effort. It's not a vaccine that you make only once and then you can use it all the time. You really, until we have a universal vaccination in terms of antigens that can elicit very broad responses, you need to update your vaccine. And so this is the work that we do every year. We continuously monitor what variants are circulating by looking at databases, wastewater surveillance, and also traveler surveillance. And this gives us a sense of what variants are circulating out there. And then we can use our own assessment. We have developed a machine learning toolkit that allows us to say if the new variant is circulating, as the chances of escaping to neutralizing responses elicited by the previous year vaccine. This is together with the epidemiology, basically how much the new variant is spreading around the world and how quickly is growing, we can start making assessments and guesses on what vaccine or what viruses we have to protect from next year. So at that point, we start generating pseudoviruses and we select a vaccine candidate. And we start basically manufacturing a risk and a number of in vivo studies in mice and eventually in humans. this data is really what we then provide to regulatory agencies across the world to approve a vaccine for next season. So the vaccine that we have developed now for this year is based on the LP81 variants. And these are just mice data, but just to point out that this new vaccine basically elicits neutralizing responses, stronger neutralizing responses against different variants. And then we also compare this vaccine versus previous year's vaccines in neutralization of different variants. And what we can see here that the new version, this is in the 1283 background, has higher efficacy than, in terms of neutralizing responses than previous year's vaccines. We do exactly the same work also for 1273. So for the both vaccines are now approved in the US. So we looked also at T-cell responses, and I just want to point out here that, when you look at CD4, we see, the strongest response against the S2 region of the molecule. And then when we look at CD8 T responses, most are really against the S1 region of the molecule. But we see T-cell responses that are broad in terms of covering different SARS-CoV-2 variants. We recently tested the vaccine, and I don't have a slide for that, in the 1283 virus vaccine, in humans in a phase 1 study. And we have shown a 30-fold booster in the 65 plus subjects, suggesting that this vaccine will provide meaningful protection for this year. So now I'll switch gear and show some of the work that we're doing on other seasonal virus, influenza. And of course, having shown success with the technology against COVID-19, flu was the next, natural target. And our vaccine is basically again on the delivery of mRNA, and we're using 3HA, mRNA encoding for 3HA, A1, the H3 and B. And really the advantage here and potential advantages that we can really have a better match between what is circulating and in terms of HA and the vaccine. And this is, in the future, potentially making those decisions about strain sequences later during the year. And this is also taking, the mRNA has shown high efficacy in older adults. So this could be also provide additional advantages. But this is, it's really the work that I will present today is on the seasonal vaccine, mRNA 1010. But of course, we think that in the future, this vaccine can also be improved. And one area, and we already have performed some clinical studies, is adding, for instance, a neuraminidase. It's more conserved antigen and has been suggested to potentially provide additional protection. And also adding additional H3 to the seasonal vaccine, to the trivalent version of the vaccine that we have right now. So I'm not going to go through the whole development of the vaccine. Actually, we did several phase 3. And during this process, which brought us to the phase 3 P304 that I will describe today, we made improvements both in the technology, the mRNA design, but also in the antigen per se, and for instance, on the B. And this really shows how quickly you can adapt. The technology allows you to really make improvements as you're doing your development. Something really hard to do, with some other technologies. So we were lucky that there were really a huge wave of influenza last year. So this allowed us to have efficacy study within one season. And in the phase 3 P304, we compared the 1010 with the licensed inactivated seasonal influenza vaccine standard dose and roughly 30,000 subjects per arm. And I want to point out that also most of the subjects, more than 50% were above 65 years of age and more than 10% above 75 years. So we looked at the efficacy and we really were pleased by the results. We defined in 3 levels of endpoints, either non-inferiority, superiority, or highest superiority. And what we observed is that the point estimate was 26.6 with a lower boundary of 95% confidence interval above 9%. So this was the highest superiority that we were aiming for, putting this vaccine in the really enhanced category. Although the study was not designed to look at the efficacy for the different circulating viruses, what we see is that really we seem to have a high efficacy for all the components, so H1, H3, and B Victoria. This is also reflected in the immunogenicity with the superior neutral, HAI titers in terms of GMR, and also in terms of seroconversion with a higher percentage of subjects seroconverting after vaccination. One question that comes often around mRNA vaccines is the durability of the response and protection. This is now, so I took the opportunity, this was not for the phase 3 or 4, but for another phase 3, where we had the quadrivalent composition of the vaccine. But we compared it to a high-dose Fluzone vaccine. So this is the enhanced vaccine for 65 plus that is approved in the US. And you can see that really the HAI titers, the trend in decline and responses are very similar. So in terms of decline of antibody responses are similar to other platforms. We have now accumulated a lot of data from different vaccines, mRNA vaccines that we have developed, and we see that the durability is very comparable. So I'll go very quickly on pandemic flu, just to say that, of course, having the seasonal vaccine, then the 1010 now is not approved yet, so we are of course applying for, we're submitting the BLA this year to hopefully have it approved the next year. But to use the technology to protect against potential pandemic flu, you need to have the seasonal vaccine approved. So, but still we have started work on the against H5. I mean, many of you have heard around, in the last 4 years about a large number of outbreaks of H5 in birds with more than 140 million poultry dead or killed. And so there's has been signs of mammalian transmission, but yet not a human-to-human transmission. So the vaccines are the clade 2.3. I mean, the strain that's circulating is this clade 2.3.4 and basically that's the sequence that we are using for vaccine development. And in collaboration with the CDC, we have shown that in ferrets, that is the preferred species, we have shown strong HAI titers after 2 doses compared to a subunit vaccine. And then in a challenge study against the virus of the same clade, complete protection from challenge. We also see decrease viral titers in the nose, so showing some potential protection from transmission. We have conducted a phase 1 study. I'm not going to describe the details of the study, but I just want to focus on the bottom part where we have basically tested an mRNA vaccine that encoded for HA, is a chikungunya strain, and 3 different doses. And recently we disclosed the HAI titers, and the only thing I want to point out is that just already after 8 days we see HAI titers, and really the response peaks at day 29, and we are above the 1 to 40 that is considered the protective threshold for flu vaccines. So I'll take the last 1 minute to go through, the next thing to do. Of course, we have developed a COVID vaccine and a flu vaccine, and the question is, can we combine them? And this might have advantages in terms of compliance. So we have conducted a phase 3 study. This is an immunogenicity study where we compared 1083 given us together with placebo to Fluzone high dose and Spikevax, or in the 50 to 64, 1083 plus placebo compared to the standard dose plus Spikevax. And really what we have seen is that in terms of the antibody response, HAI titers, we are, for at least the 3 strains that are in the current flu vaccines, we have superiority. And the same is true for the, of course, the COVID-19 component. We are, in this vaccine, we have 1283 and not 1273. So basically the vaccine seems to have the right immunogenicity and non-inferior to both Fluzone and Spikevax. And so we are in discussion with regulatory agencies about the regulatory path for approval. So I will finish by showing that basically we have developed a number of vaccines. Today I focus on the respiratory ones. We have vaccines against latent infection, herpes viruses, but also we have developed vaccines against flaviviruses and also norovirus and Lyme. There is, of course, a great effort also in the oncology space, where we have some promising phase 2 data, and also in rare disease, where a metabolic disease where you can provide mRNA to express the protein that is defective in some patients. And with that, thank you, and I'm happy to take any questions.
What is the thermal stability of flu mRNA vaccine from Moderna?
Stability in terms of the vial
Storage condition, yeah.
Yeah, this is depends on, it changes with the country, and so I think right now we are 3 months, 4 degrees, and then more with -20. This is it's a good point. I mean, it's continuously evolving, so we think that we can get to 7 to 1 year stability of 4 degrees. But this is, of course, it's part of the discussion with regulatory agencies.
Thank you.
This was a problem during the pandemic. I think it's part of developing the technology and better understand what is needed.
Thank you.
Andreas, thanks. Having a better match to flu would be a benefit by itself, right? So how close to the season can you come?
Yeah, I mean, I think that's an excellent question. So, you know, to give you an example, I mean, in the last, not the last year, but the years before, because things have changed a little bit, but we were able to really make the vaccine in May, June. So you can get relatively close.
That's good.
The challenge is more, again, regulatory hurdles and when you have to submit all the documentations and what are the requirements. So we are actually going right now, we're going back so that we are start making those choices, you know, earlier. But there is interest, I would say, from some regulatory agencies to start having those discussions on when, you know, how late this could happen. And I think it will depend also on, until we are the only, you know, or 2 mRNA vaccines company, I think that's more challenging, but as there are more transition to mRNA, will become more accepted.
And I can't let you get away without asking for a comment on the political environment mRNA vaccines find themselves.
Yes, it's I think there is my answer to that is that there is a lot of education that needs to be done in the context of mRNA vaccines and what they do and what they don't. It's in part is on us, to be honest. In part is, of course, also in the scientific community. The positive, I mean, I'll try to be on the positive side. 1283 was approved this year, RSV 18 to 59 as well. So at least with additional efforts, we are getting through certain things.
I have a quick question. Do you have a sense of how many mRNAs can be combined before you start losing immunogenicity?
Yeah, I don't think there is a limit. I mean, in clinical studies, we've done up to 6. In preclinical, we're gone up to 15, but just because that was the target. You don't need to have all the mRNA in the same LNP. Multiple LNP will transfect the same cell. So you can, you know, that allows you for a lot of flexibility.
Thank you. 2 practical issues. One, there's very little on which side we should get the COVID each year. There's some preliminary studies suggesting it's best to get it on the same side, but very small data. So that's the first question. Second, us elderly people get many vaccines, including respiratory syncytial virus vaccine, younger kids trying to get an HPV vaccine. What do we know? You started talking about combination. What do we know about the safety of giving more than one at the same time? Or if not, how should we space them?
Yeah, I mean, we have done, there are multiple, you are raising multiple questions. In terms of where to give it, I mean, I think that now that everyone has been, I mean, most of the people have been vaccinated, or I think you can give it to the same. I mean, it doesn't really matter at that point. You know, as initial vaccination probably would go to the same site. Then I think you know, combination is appealing. I think, you know, especially if you think about the pediatric population, where multiple vaccines and multiple injections are needed and multiple visits, if you can combine, combinations have been very successful in the pediatric. I think as we develop more vaccine for older adults and the population is aging, I think it will be important to, you know, combine some of those vaccines to decrease actually, again, the number of visits. We have shown, of course, that this is a part of the regulatory approval and that these vaccines can be co-administered also with existing vaccines without a significant impact. I mean, you have to match non-inferiority, so there's no impact really on immunogenicity from that perspective. You have to show that. No, there's no, we have no evidences for that, right? I mean, in some cases, it's a, I mean, this points to something else. I mean, for vaccines, when you combine them, sometimes you start seeing interference. So the response towards one of the components of the vaccine decreases, right? And this is shown also for other technologies. We have seen it, I mean, of course, you show the positive stories always about the combination vaccine for flu COVID. Initially, we had the Spikevax, so the first generation, and we saw interference. So then we changed it to the smaller version of the vaccine, and that's just a problem. So interference is something that you have to look for. Yeah. Ciao.
Oh, one more.
Thank you. One of the factors that has slowed a number of companies in developing an mRNA flu vaccine has been poor immunogenicity of the B strains, flu B. I'm curious what it took to generate the pretty good looking flu B data here.
Thank you for that question, and it reminds me that I should always have some, you know, the research side of things. So yes, so that's true. And so, you know, I showed the different phase 3 studies, and what happened in one of the first studies, we had a lower immunogenicity on the B strain. So we had to re-engineer the molecule to increase the stability and expression of the B to get to, you know, to the acceptable immunogenicity. So I think there is a lot of potential in improving these vaccines also through antigen design in the future.
Hey, thank you so much.
Thank you.
I think we'll